All right, welcome everybody. It's Friday, March 20th. Welcome to the last day of the first week of online school. Hopefully it wasn't too miserable for you. I feel like it went relatively well. It seems like everybody was keeping up with their work for the most part. If you are not, now would be a good time to make some of it up. I know I said that there was a 0 policy for late work, but I've been a little bit lenient with the first week. I won't be lenient in the second week. So please make sure if you're going to make stuff up, you get a hold of me that you're going to, and let me know you're going to do that. Otherwise, they will stay as zeros. If they are zeros, you've probably already noticed that they are seriously affecting your grade, and you can go entire letter grades in a few days. So please make that stuff up. Okay? So we have finished meiosis and mitosis, and we're moving on to Mendel. So we're talking about Punnett squares, and all of the stuff that goes on in with Punnett squares. So alleles, allele frequency, phenotypes, genotypes, stuff hopefully you've done before, and it'll be a little bit of a review for you. I know some of you have said you liked Punnett squares. Hopefully this won't be miserable. All right, so let's go ahead and jump in. All right, before I get started talking about Mendel, I just want to talk about some of the historical beliefs. What did people believe about heredity before Mendel? So people did realize that their offspring looked like the parents. They used the phrase like begets like. So two parents will produce an offspring that look similar to the parents. However, some people did believe that it came from the mother completely, so females controlled all the traits. That's what maternal heredity is. It comes from the mother. And then you had your people who thought that it was paternal heredity, so it comes from the father. So all of the traits would come from the male, regardless of what the child looked like. Now, they didn't think that they were exact clones, and obviously they couldn't. If you looked at a son and a father, they're not going to look exactly the same. But they did believe that all of the traits came from one of the parents, except for a select few who thought that it was a blending. So the offspring were the average of both parents. Now, that might sound right, but you have to keep in mind that when a child looks like the mom or looks like the dad, they don't have 100 or 50% mom's DNA and 50% dad's DNA. You can have more traits from your father's side than your mother's side, so it's not exactly an average of both. And then you had your last group of people who believed in something called a homunculus, and this was the idea that the sperm of the male contained fully formed little people, microscopic people. And you'll see this on the next slide. If you look at the drawing here, you can see that this is what they thought. So there's an actual person inside the sperm. And there's a Swedish text from 1640, and they called it Little Man. They called this homunculus, this little person in there, Little Man. And some even earlier textbooks before this said that they had men who were fully clothed. So imagine that, a fully clothed person inside of a sperm. So obviously there were some pretty wild thoughts out there, but what we know now, or what Mendel proved, changed the worldview very drastically. All right, so before we get started on Mendel's work, let's just talk a little bit briefly about who Mendel is and what he did. You don't have to write everything on this slide. I would write just what I highlight. It'll save you a little bit of time. All right, so Gregor Mendel, he was born in 1822, and he would pass away in 1884, so he lived to the age of 62, which not terribly old, but pretty good for the time. He's considered the father of genetics, so that's probably a pretty good thing to write down. It's kind of a nickname for him. He's the father of genetics. He grew up on a family farm in Austria, which for those of you who don't know, it's right near Germany. But he gardened, he studied beekeeping, but he was a sickly child, so he couldn't take over the family farm, and because of that, he went to school, and he studied philosophy. All right, now they didn't really have student loans back then, so in order to pay for his education, he became a friar, a religious man. He entered the Augustinian St. Thomas Abbey, and he trained to become a priest, and he taught science. So he did two things, and he taught science at a university level, so he was a very bright man. He taught both physics and biology, so he was well versed in more than one science. After he did all of his work, he actually returned to the abbey, and he taught while studying heredity. So he was a teacher, and all of the experiments that we're going to look at is during this time when he returned to the abbey. After he did his work, he became an abbot, which is the head of an abbey, and he gave up science. Boo, but he did. He really had to focus on his responsibilities as the abbot. All right, so that's Gregor Mendel. The rest of this chapter is going to deal with his work and what he discovered and why it was important. So Gregor Mendel primarily worked with pea plants, and there was several different reasons why he did that. The first one is they had many what are called dichotomous traits, which just means you either have this variation of the trait or the other. There's no blending, so you either have a tall plant or a short plant. There's no medium plants. You either have a green seed or a yellow seed. It's a dichotomous trait. It's one or the other. There's only two choices for what they can be, so it makes it really easy in determining which trait the offspring has. Many are called traits were true breeding, and don't worry too much about that now. We'll talk a little bit more about that later, but pea plants are true breeding. They are monoecious. This is a word you should hang on to because it'll come back. Monoecious. It means they have both male and female parts in one flower. So this is important because you can cause the plant to basically have sex with itself, so it'll fertilize itself. And the reason it can do that is because if you take a look over here at this picture here, the plant. On the plant, same flower, you have an ovary, which is the female part, but you also have an anther, which is what's going to contain pollen, and pollen is just male reproductive cells. So you can akin pollen to sperm. So if they're on the same plant, this pollen that is right here can make its way to what's called a stigma, travel down the stigma where the ovaries are at, and pollinate or fertilize itself. So you can what's called self-fertilize the plant. And that again is really important. And when we start talking about the generations of plants, that will come back. They have short generation times. This is something that's really important in heredity studies still to this day. You have to pick organisms that reproduce quickly. So it'd be really hard to do a heredity study in humans because the average human lifespan is 76 years. So your subject could outlive your scientist. So you have to pick organisms that live for a short amount of time and reproduce quickly. And then lastly, they were cheap. They were very cheap for him to get tons of, and they were readily available. So there was lots of them. So that's why he chose pea plants. All right, so let's talk a little bit about the notation system that Mendel used. How did he identify things? It's pretty easy. If you see a pea, that is the parent generation, so the generation you're starting with. The F1 generation is the first generation after the parental generation. You have the F2 generation, which is the offspring of two different F1 generations. And then everything after that, you just continue on with the numbering. So F3, F4, F5, so on and so forth, as many as you did. All right, siblings, you know that. They're just brothers and sisters. Cross sibs is when you mate between brothers and sisters, which obviously you don't see very much in the animal kingdom, but you will see it in plants. A back cross is when you take an offspring, so one of these F1 generation, and you mate it with one of the parents or maybe even an older ancestor. A self-cross, and this is one I'm going to highlight in a different color because it's something you're definitely going to see in this, is when you take a plant and you cross it with itself. So if you don't know what I'm talking about, go back to the previous slide. Look at self-fertilization in plants. That's why we went through how a plant can fertilize itself. So go back to that if you have to, but self-crosses are important. And I also told you we would go over true breeding. So here is true breeding. It's when the parents have the same traits. When parents having the same traits always produce offspring that have the same traits. So if you had a true breeding flower for green seeds, So there's your green seed. Every offspring of that parent will have green seeds, no matter what. Okay, so that is a true breeding plant. And that's a horrible drawing, and you may not even know what I'm doing there because I, first of all, just used red. But that's a true breeding plant. So every offspring of that parent will have green seeds. So same trait always. All right, so now you know why he used pea plants, that he used pea plants. You know some of the notation he used for his experiments. Now let's talk about what he did. So we'll use an example here. One of the things he noticed, we're starting with a true breeding purple plant and a true breeding white plant. And if you remember back, this little P here designates that this is the parental generation. So we know that's the generation we're starting at. And the other important thing to note is both of them are true breeding for their trait. So we've got true breeding white flowers and we've got true breeding purple flowers. Okay, so if you were, in theory, to reproduce this white flower with another white flower, you'd only get white flowers. If you were to reproduce this purple flower here with another purple flower, you would only get purple flowers. So that's our parent generation. What did Mendel find? He found that all the resulting F1 generation, so those first offspring of the parental cross, were all purple. Oh, that's terrible. Hold on one sec. All purple. Now, this was really surprising to Mendel because up to this point, Mendel only thought that he would get a blending. So he'd get some light purple flowers. So this was a very big surprise for him. The fact that there was no blending was a significant discovery. So what did he do? He went and crossed these two with each other. So he crossed two individuals of the same F1 generation. He could have self-crossed, so he could have crossed one flower with itself. But two of the F1 generation, what he found was you would have 3 purple, and you'd have 1 white. I'm not going to write that in white because that wouldn't make any sense to you. You wouldn't see it. So he found a 3-to-1 ratio, 3 purple plants to 1 white flowered plant. This was kind of what jump-started his whole work. He was very surprised by this, mainly because there was no blending. This was very surprising. Alright, so this slide is a pretty good summary of what I talked about on the last one, but I did leave something off. The term monohybrid cross is a cross between monohybrid. That's just a cross between 2 organisms with different variations of one trait. So flower color is what we looked at last time. You can have dihybrid and trihybrid crosses. That just means you're looking at more than one trait. Right now we're only looking at flower color, so it's a mono, meaning one hybrid cross. So in one experiment, like we just said, Mendel crossed true breeding violet flowers with true breeding white. The F1 generation, which is here, had all violet flowers. If you self-fertilize the plant, so if you take this plant right here that's got an arrow pointing to it and fertilize it with itself, you get the F2 generation, which is right here. And he found that in the F2 generation, you had a 3-to-1 ratio. Remember that ratio, 3-to-1. Because we're going to see it with all these different traits when we do these kind of crosses. So for a formal monohybrid cross, which is what we did right here, you get a 3-to-1 ratio. And we saw this in all the traits. He looked at seed color, plant height, flower color, wrinkled versus smooth seeds. All of these crosses, he ended up with a 3-to-1 ratio, so it was pretty significant. So Mendel was able to draw some conclusions based on the crosses he did between pea plants, so we're going to go through them now. The first one was that inheritance appeared to be particulate, and what he meant by that was that particles are passed from parent to offspring, and these particles carry genetic info that would lead to a specific characteristic. Now we know today that these particles are actually called genes. So what he would refer to as particles, we now call genes. But that's what he meant by inheritance appeared to be particulate. They were just passed down, again, by genes. He also noticed that some versions of a trait mask other versions of a trait, and we refer to this as dominant. So if we have a trait for eye color, okay, let's just say eye color, and you can either have, let's say it's in fruit flies, you can either have red or white. Alright, so if we were going to write the genes for that, you'd have capital A for red and little a for white. So any fly that has red eyes is going to have a big A and either another big A or a big A and a little a. But if it has that big A, it will have red eyes because big A is dominant. Okay? Opposite of that, he found that other versions of a trait were masked. They were called recessive. Alright, so lowercase letter is going to indicate a recessive allele. So in this instance, the only time you will have a fly with white eyes is if you have little a, little a. It's the only time you'll end up with a recessive trait shown. The recessive trait appeared to have passed through the F1 monohybrids unchanged to the F2. So what that means is when you take big A little a and you cross it with big A little a, and this is in the F1, you're going to get an F2 that has big A, big A, big A little a, and little a little a. So the recessive trait was still there even though it wasn't shown in the F1. It was still present in the F1. This is where your 3-to-1 ratio comes in. So what he saw was after you did a monohybrid cross, where you cross big A, little a, and big A, little a, you get 4 individuals. You get 1 big A, you get 2 big A, little a, and you get 1 little a. So if you think about phenotypes, if you go back to thinking purple plants and white plants, this is a purple plant, this is a purple plant, this is a purple plant, but this is a white plant. And that's your 3-to-1 ratio. You have 3 purples to 1 white. And this happens because that recessive allele is still present, okay? Even though it's not expressed in the F1, it's still present. So this would be your F1, this would be your F2, okay? I don't have anything to say. You can just exit out that last one and keep it out of your notes so it's not blocking my writing. Okay, so this is a really text-heavy slide. So what I would do is maybe, create like a Quizlet, something with these words on it too, so you can practice, because they're all just vocab words, and they would be on some sort of matching section on a quiz or a test. So make sure you can identify these. Let's quickly go through them. I'm not going to sit here and read all of them to you, but you need to be familiar with them. So the first one being a trait, that's just a characteristic. So flower color, eye color, seed, texture, plant height, whatever. It's just a physical characteristic. A phenotype is the characteristic version that we see. So eye color. So if we were looking at phenotype, a phenotype would be blue eyes or brown eyes. All right, green eyes. It's actually what you see. Okay. I'm going to skip down to genotype because genotype and phenotype are different. So genotype is the actual genes. It says hereditary particles. That's what Mendel called them. We're going to go ahead and start calling them genes. That's the actual genes that are present. So big A, little a, little a, little a, big B, big B. It doesn't matter what letters you pick. It's just the fact that you're showing one dominant, one recessive, or both dominant, both recessive. It's just the actual combination of genes that are there. And these specific types of genes are called alleles, which we see here. So they're the actual versions of the genes. We've already talked about dominant, recessive, homozygote versus heterozygote. Remember that homo means same. So it's when you have two that are the same allele. So big P, big P, little p, big A, little a. Again, whatever letters, they're the same. They're either both capitalized or both lowercase. And that's different from a heterozygote, hetero meaning different, and that's when alleles have the same, or alleles for the same trait differ. So you're always going to have one capital and one lowercase. Okay, those are heterozygotes. And then last, the zygote just is the first cell after fertilization. So your zygote is eventually going to form the embryo and eventually the baby. So we've got some more definitions here. A gene is a place in the DNA that encodes for a trait. So you have a gene for, like I said, hair color. You have a gene for eye color. You have a gene for height. You have all kinds of genes in your body, and those are just where on the DNA these alleles are present. A locus is a place of interest on a chromosome. We're usually looking at a gene. I'm not really 100% concerned with you knowing that. We've already done alleles. Not sure why it's on there twice. I guess it's that important. I would say it is. A reciprocal cross is a form of cross or a form of mating where a previous cross is repeated, but the parents exhibiting versions of a phenotypic trait are reversed by sex. Again, I'm not 100% sure that you all need to go over that. It was just a way for Mendel to eliminate all of these traits being dependent upon the gender of the plant. Again, for you all, don't worry too much about it. The wild type allele, I do want you to know. Wild type allele. It's just the most common allele in a population, so it's what you would consider the normal allele. It's usually people associate it with the dominant allele, but it's not always. Then you have a mutant allele, which is an allele that's rare in a population. Okay, rare in a population, so it's reason to be the most recently formed allele by a mutation, but that's again not always the case, but it's a reasonable assumption you can make. Alright, so we're already at 22 minutes, so this looks like a good place to stop. We'll keep a little shorter today. Hopefully you're taking good notes and filling everything in. We'll pick up on Tuesday with Mendel's first law, and we'll talk more about Mendel's work. Make sure you get your notes turned in by 8 a.m. 8 a.m. on Tuesday. Please do it.